Every year, a stomach virus rips through cruise ships, school cafeterias, army barracks, and family dinner tables, and it seems to grab almost everyone in its path. Except it doesn't. On the exact same ship, eating the exact same food, sitting next to people who are throwing up for 2 days straight, some passengers simply never get sick. Not a headache, not a cramp, nothing. For years, doctors wrote this off as luck. Maybe they washed their hands better. Maybe they ate less shellfish. Maybe their immune system was just having a good week. Then scientists started digging into the DNA of these unlucky turned lucky people, and what they found flipped the whole story upside down. It was not luck. It was not soap. It was a tiny switch buried in their genetic code, one that some people are born with and some people are not. And it turns out to be one of the strangest and most powerful examples of natural selection still working inside the human body right now, in real time in this century. This is the story of how researchers figured out why some people are almost completely immune to one of the most common stomach viruses on the planet, and what that discovery reveals about our past, our biology, and the strange bargains evolution makes to keep a species alive. If you like stories about how our DNA quietly shapes our lives without us ever noticing, stick around, because this one gets stranger the deeper you go. Let's start with the virus itself, because you cannot understand the gene without understanding what it is up against. The bug responsible for most of this misery is called Norovirus. You have probably heard it called the stomach flu, even though it has nothing to do with influenza. It causes sudden vomiting, cramping, and diarrhea, usually starting less than 2 days after exposure and clearing up within another 2 or 3. It sounds mild on paper, but norovirus is brutally efficient. It takes as few as 18 viral particles to infect a person, a number so small it is almost impossible to picture. A single vomiting episode can release billions of these particles into the air and onto surfaces. It survives on countertops, doorknobs, and food for days. It shrugs off many disinfectants that would kill other viruses without breaking a sweat. Public health researchers consider it one of the most contagious pathogens ever studied, responsible for roughly a fifth of all diarrheal illness worldwide and hundreds of thousands of deaths a year, mostly among small children in places, with limited access to clean water and medical care. Because it spread so easily, norovirus outbreaks became a strange kind of natural experiment. Put 400 people on a cruise ship, expose them all to contaminated food or water, and normally you would expect nearly everyone to fall sick within days. But outbreak investigators kept noticing the same odd pattern over and over. A meaningful chunk of the exposed group, sometimes as much as a fifth of everyone on board, never got sick at all, even though blood and stool tests later confirmed the virus had definitely entered their bodies. Their immune systems were not fighting off an infection. The virus simply could not get a foothold inside them in the first place. This mystery caught the attention of researchers in the late 1990s and early 2000s, who ran a series of what are called human challenge studies. In these tightly controlled experiments, healthy adult volunteers agreed to be deliberately given a measured dose of norovirus, so scientists could watch exactly what happened next. It sounds unpleasant, and for the volunteers who got sick, it absolutely was. But the results were extraordinary. Roughly 70 to 80% of the volunteers developed symptoms as expected. The rest did not get infected at all, regardless of how large a dose of virus they swallowed. Researchers tried increasing the amount of virus given to the resistant volunteers, assuming they simply needed a bigger push. It made no difference. Something inside their bodies was acting like a locked door that no amount of knocking could open. At first, scientists assumed this had to be an immune system difference, maybe leftover antibodies from a previous exposure to a similar virus. But when they tested the resistant volunteers, many of them had no prior antibodies at all. Whatever was protecting them was not something they had learned to fight off. It was something they were simply born with. The breakthrough came when researchers started looking not at the immune system, but at the surface of the cells lining the human gut, the exact spot where norovirus first tries to attach itself and break in. To infect a cell, a virus generally needs to dock onto a very specific molecular shape on the outside of that cell, almost like a key fitting into a lock. Scientists discovered that norovirus uses molecules called histo-blood group antigens as its docking point. These are sugar structures that sit on the surface of cells lining the small intestine, and in many people, they also show up in bodily fluids like saliva, tears, and mucus. Here's where it gets interesting. Not everyone produces these sugar molecules in their gut lining. Whether you do or not comes down to a single gene called FuT2, short for fucosyltransferase 2. This gene carries the instructions for building an enzyme that attaches a particular sugar tag onto the surface of certain cells, including the ones lining your intestines. People with a working copy of this gene are called secretors because that same enzyme also causes blood group antigens to show up or be secreted into saliva and other body fluids. Roughly 8 out of every 10 people on Earth fall into this secretor category. But a meaningful minority of the human population carries a mutated version of the FUT2 gene that switches the enzyme off entirely. These people are called non-secretors. Their gut lining never displays the sugar structure that norovirus depends on to attach itself. Without that docking point, most strains of the virus simply cannot get inside the cell to begin an infection. It is not that their immune system fights off the virus faster. The virus never successfully attaches at all. It passes through the body like an unwelcome guest, who cannot even get through the front door. When researchers compared the DNA of the resistant volunteers from those human challenge studies against the people who got sick, the pattern was almost too clean to be believed. Nearly every person who resisted infection was a non-secretor. Nearly every person who developed symptoms was a secretor. A study published in a leading medical journal in the early 2000s confirmed it directly, and later research across different countries and different norovirus strains kept finding the same connection. One single gene, doing one single job, was the difference between a miserable week and a complete non-event. Naturally, the next question researchers asked was how common this protective mutation actually is and where it came from. The numbers turned out to vary enormously depending on where in the world you look. In European populations, non-secretors typically make up somewhere between 15 and 20% of people. In parts of Africa, the number can run higher. In some East Asian and Pacific Islander populations, the mutation is far rarer, sometimes under 5%, while in certain isolated communities it barely appears at all. This is not random noise. It is the fingerprint of natural selection, acting differently on different populations, depending on which diseases those populations were fighting off for thousands of years. That last point matters, because FUT2 does not only affect norovirus. It turns out this one gene sits at the center of a much bigger story about infectious disease and survival. The same sugar structures that norovirus uses as a door also happen to be used in different ways by several other pathogens. Certain strains of rotavirus, another major cause of severe diarrhea in young children, rely on similar attachment points. Some researchers have found associations between secretor status and susceptibility to particular strains of cholera, and to some respiratory infections as well. In other words, the FUT2 gene is not a single-purpose lock. It is more like a master key slot that a whole range of different microbes have independently evolved to exploit, each one for its own reasons. This is why scientists think the mutation that shuts down FUT2 never fully swept through the entire human species, even though it clearly offers real protection against a genuinely dangerous and widespread group of diseases. Being a non-secretor helps against norovirus and some strains of rotavirus, but studies have also linked non-secretor status to a higher risk of certain other conditions, including some types of urinary tract infections and a modestly increased risk of Crohn's disease, an inflammatory bowel condition. Meanwhile, being a secretor appears to carry its own set of trade-offs and small advantages against a different lineup of threats. Evolution rarely hands out a mutation that is purely good or purely bad. More often, it hands out a trade, protection against one danger in exchange for vulnerability to another, and which side of that trade wins out depends entirely on which diseases were actually killing people in a given place at a given time in history. Geneticists have tried to pin down roughly when the non-secretor mutation first appeared and started spreading. The estimates point to a mutation that arose a very long time ago, likely tens of thousands of years back, and then rose in frequency in certain populations because the people who carried it were more likely to survive childhood, more likely to have children of their own, and more likely to pass that same protective version of the gene down the line. Multiplied across hundreds of generations, a small survival edge against a killer as efficient as severe diarrheal disease is more than enough to shift the frequency of a gene across an entire population. Before modern medicine, before oral rehydration therapy, before clean water infrastructure, a bout of severe vomiting and diarrhea in an infant or a small child could easily be fatal. A gene that meant your gut simply could not be infected by one of the most common causes of that illness was not a minor perk. It was potentially the difference between a family line continuing or ending. There is a particularly striking piece of supporting evidence that comes from looking at the genome of an entirely different group. Neanderthals and Denisovans are extinct close relatives. When researchers sequenced ancient DNA extracted from Neanderthal remains, they found that Neanderthals appear to have carried working, active versions of the FUT2 gene, meaning they were secretors, at least in the specimens studied. This tells scientists that the non-secretor mutation seen in some modern humans today likely arose within our own lineage after we split off, or possibly through a completely separate mutation event that happened more than once in human history. Scientists have actually found evidence that the FUT2 gene has been independently knocked out by different mutations in different populations around the world, a phenomenon called convergent evolution happening at the level of a single gene. Populations that had no contact with each other, separated by oceans and continents, each stumbled onto their own version of the same solution because they were facing similar evolutionary pressure from similar gut pathogens. Doctors and public health researchers have since put this discovery to very practical use. Knowing whether someone is a secretor or non-secretor has become genuinely useful information in a few different areas of medicine. Vaccine researchers working on norovirus vaccines have had to account for secretor status directly because a vaccine trial that does not track this gene risks badly misreading its own results. If a vaccine appears to protect a group of volunteers, but that group happens to include a larger than average share of non-secretors who were never going to get infected anyway, the vaccine can look far more effective than it actually is. Understanding Fu-T2 status has become a standard part of designing and interpreting these trials properly. Doctors treating unexplained recurrent urinary tract infections or certain digestive conditions now sometimes consider secretor status as one piece of a larger puzzle, since the same biological switch shows up in research connected to those conditions as well. And on cruise ships and in hospital wards, epidemiologists investigating a norovirus outbreak already understand, without needing to test anyone's DNA directly, that a certain percentage of exposed people will simply never get sick, and that this is not a flaw in their contact tracing. It is baked into human biology itself. What makes this whole story land so hard is how invisible it is in everyday life. You cannot look at someone and tell whether they are a secretor or not. It shows up in no physical feature, no skin tone, no eye color, nothing you could ever guess by looking at a person standing in front of you. It lives entirely inside a few letters of genetic code that quietly determine whether a virus that has ended up wrecking an entire wedding reception, an entire naval ship, or an entire elementary school will find a foothold in your gut or simply pass through unnoticed. Two siblings raised in the same house, eating the same food their entire childhood, can carry completely different versions of this gene and live out completely different relationships with something as ordinary as a stomach bug going around. It also forces a bit of humility about how we think about health and immunity in general. It is tempting to credit a strong immune system, a clean diet, or careful hygiene whenever someone dodges an illness that took down everyone around them. Sometimes that credit is deserved, but sometimes the real explanation has nothing to do with willpower or lifestyle at all. It is a molecular lock on the surface of your intestinal cells that was set in place before you were born, inherited from parents who inherited it from their parents, stretching back through an unbroken chain of ancestors who survived their own encounters with gut pathogens long before anyone had ever heard the word virus. There is something almost poetic about the scale of it too. An entire global industry of hand sanitizer, disinfecting wipes, and quarantine protocols exists to fight off a threat that, for a meaningful slice of humanity, was never a threat to begin with. Not because those people are careful, but because of a single letter change in a single gene that happened to occur somewhere in their family tree tens of thousands of years ago on a continent they may never have set foot on. In response to a disease pressure none of them will ever personally remember. Scientists are still working out the full picture. Newer strains of norovirus have already been found that can partially get around the non-secretor barrier in some cases, which is a reminder that evolution never really stops. The virus is under just as much pressure to find a new way in as the human gut is under pressure to keep the door shut. It is an arms race that has been running quietly for tens of thousands of years and is still running today, invisible, unfelt, but written directly into the DNA of nearly every person on this planet. So the next time you hear about a stomach bug tearing through an office, a school, or a ship full of passengers, and someone shrugs and says they just never seem to catch these things, there is a real chance they are telling the truth in a way they do not even realize. It might not be their diet. It might not be their immune system working overtime. It might simply be a switch flipped off in their DNA a very long time ago, quietly doing its job, one meal, one outbreak, one generation at a time.